Hi, so my name is Shamika,
And I'm Adelaide.
and we're here to talk to you about DNA sequencing. DNA sequencing is used to map out the sequence of nucleotides that make up a strand of DNA. So it was first done by this guy, Frederick Sanger, in 1977, and he later received the Nobel Prize in Chemistry in 1980 for his contributions concerning the determination of base sequencing in nucleic acids. His method is known as the Sanger method, but it's also known as dideoxy sequencing or chain termination.
Okay, in order to do the chain termination method, one must first obtain the DNA of interest. The DNA is then denatured by heat and separated with the template and the complementary strand. Um, One must then obtain a primer and attach it to the 5' end of the DNA. 4 flags are obtained and added to each flag are the temporary strand, the primer, DNA polymerase 1, and DNTP. Basically, the DNA polymerase helps make the DNA sequence, and the primer helps start it. DT, a DDNTP is added at the end of the flags to help with DNA termination sequence. A pipette is then used to draw from each flask and laid on gel electrophoresis. I got it right this time. So basically, the DNA is added on the negative end of the gel electrophoresis and goes down to negative onto the positive charged area, because we all know that DNA has a negative charge. After this is done, we're gonna analyze the bands that are created, and by looking at the bands from each lane ACTG, one can come to a conclusion as far as what is the particular sequence of the DNA of interest that we're looking at.
Time, labor, and cost. So this method does take a lot of time. The Human Genome Project, which started in 1990, took over 13 years to decode using this method. It basically takes over a year to sequence 1 gigabase. For the Human Genome Project, it took $3 billion, but this was for research and development. But the actual cost for just sequencing was $500 million. As of 2012, the cost per mega base pair of DNA sequence is 9 cents, and per genome it is $7,950.
Okay, so the advantages of using this method, well, one, is very easy to understand, and two, is one technique regardless of scale. It uses fluorescent dye for each of the 4 bases. Its fewer genomic regions are sequenced multiple times, and it requires commercially available DNA polymerase 1. The disadvantages of using this method, it requires that each restart be cloned for production of single-stranded DNA. In addition, the technique was inefficient due to the vast resources needed to compute a single genome. The sequence only one section of DNA per reaction, up to about 500 base pairs. Each new round of sequencing requires a new primer to be synthesized, which increases the cost. Last but not least, it's not completely accurate, so one must repeat sequencing more than once.
So these are the related methods. These are different kinds of methods to sequence DNA. There's chemical sequencing, automated sequencing, shotgun sequencing, and next generation sequencing. So that concludes our presentation. Thank you.